Hi everyone, so we finished section 2 on this XML formatted data showing the KEGG pathway for glycolysis. So this is one of the key types of data that we'll be looking at, biological network data. Another type of data is omics data. So omics, high throughput data. There are, again, four major categories of this: genomics, transcriptomics, proteomics, and metabolomics. This is a sort of cartoon showing the traditional biochemists that used to be studying a single gene at a time, and the new genomics, proteomics researcher who basically studies all of the genes in the organism, all the proteins in the organism, all at the same time. So you may recall from David's lectures, The Central Dogma of Molecular Biology. This was essentially this flow of information from DNA, which is stored in the DNA sequence of the genome of the organism, onto its different genes here are transcribed into pre-messenger RNA, and then we get from this RNA splicing of the introns and eukaryotes to make the final messenger RNA, which goes outside the cell nucleus, and attaches to a ribosome to be translated three nucleotides at a time, making it per codon, into particular amino acids, and form this string of amino acids which make a protein, which then folds into a final protein. Hopefully you've looked at the animations that are actually on the Moodle site showing gene transcription into messenger RNA, and also the translation of the messenger RNA into proteins. So there's two good introductory videos animating those processes on the Moodle page that are worth watching. How does our data, our omics data, relate to this? Well, the DNA sequence of the organism is the genomics data, essentially the sequence data. The transcriptomics data is the level of messenger RNA. It's actually the transcriptional activity, the expression, gene expression level of a particular gene into messenger RNA. How much messenger RNA is in that cell. And then proteomics data is the concentration of the different proteins within the cell. So this is what proteomics techniques usually measure. And then the third form, the metabolomics data, doesn't fit into the central dogma of molecular biology. It's the level of the different metabolites like glucose, etc. within the cell. So how is the actual network data that we looked at previously related to this omics data? We saw that relationship in the first lecture, showing how the gene expression data in Streptococcus pneumonia related to the activity of the different genes in that pathway, as that pathway was actually activated and turned on different genes. And it's a similar idea here. We're going to look at the insulin pathway as an example. But our genomics data is in the nucleus. It's the the DNA and the particular genes, the sequences of the particular genes making up the system which we're interested in. So in this case, these genes actually code for the enzymes which are involved in the metabolic pathway. So we've got a particular gene being transcribed, and that is our transcriptomics data. So this is the actual activity of that gene, how much of the messenger RNA is being expressed by that gene. The proteomics data gives you information about the level of the proteins. So it's not only the protein levels, but we potentially get from proteomics the state of the phosphorylation. So this can be very useful to tell how much activated IRS1 is actually in the cell compared to how much inactivated without any phosphate is in the cell. And then metabolomics data gives you the levels of these metabolites, glucose 6-phosphate, pyruvate, and glucose within the cell. So we see that we can get a complete picture, or at least some picture of what's going on in the cell using these 4 different omics technologies. So going a bit more into detail on these technologies, David's already discussed genomics quite a lot. Genomics is about determining the complete sequence of the DNA genome of an organism. This was first carried out in a very small phage virus, about 5 kilobases, 5,000 base pairs, in 1977 by Freck Sanger, who got the Nobel Prize, and the Sanger Institute in Cambridge is named after him for this accomplishment. The first free-living organism, not a virus, was sequenced in 1995, 1.8 megabase pairs, so quite a much larger organism, H. influenza. And then finally the human genome sequence was produced in around April 2003, 3 billion base pairs. So sequencing has come on quite rapidly over the last 40 years. Again, where do we find out about sequence genomic databases? Well, the Nucleic Acid Research Database summary articles give this information. If we go down to this particular link, human and other vertebrate genomes, we can do model organism genomes, and there are huge numbers of them. Bovine genome, the cow genome, but one of them which is particularly useful is Ensemble. So David may have mentioned Ensemble. This provides, so it was originally developed at the EBI, the European Bioinformatics Institute, for sequencing the human genome, but it's now, the sequencing powers, once the human was finished in 2003-ish, they moved on to other eukaryotic organisms, particularly the mouse model organism, but then now we have, 20 years later, a huge number of eukaryotic genomes, alpaca, bush baby, cod, cow, dog, dolphin, elephant. So each one provides a complete genome sequence for that organism. Going into detail a bit, chromosome 19, we've got this human genome, we've got the chromosome sort of structure itself, and if we zoom into this little section by this red rectangle, we get into this area of chromosome 19, and you can see that I've zoomed in on the ApoE gene. So that is the gene, that little sort of section here, that little red sort of rectangle here, that is the ApoE gene, whose different isoforms, E2, E3, E4, whatever sequence someone has in that gene, in that position, it gives them either a predisposition for Alzheimer's or it makes them less likely to get Alzheimer's. Just this little region of the chromosome 19. So that's the sort of data that genomics can give you. A complete 3 billion base pairs. But we can zoom even farther in to the ApoE gene, and this shows you the structure of the gene itself in terms of exons and introns. So the exons are the filled in areas which code for protein, and the introns are spliced out when you do splicing. You can see that most of the length of the nucleotide sequence, in this case, is exons. It just gets spliced out. The actual coding regions are quite small. And so this is, including information about individual genes, exon, intron structure. We can zoom even farther in. So this is zooming into a particular region of the ApoE gene. And we come to the actual nucleotide sequence itself, A, C, T, 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 etc. And for each group of 3 nucleotides, so we've got this TTT, it codes for this amino acid phenylalanine. TTT, phenylalanine. GTA is valine. So you've got 3 reading frames going that way and 3 reading frames going the other way from the other strand of DNA. So these are reverse complementary sequences. So that's the sort of detail that so you can, from the reference genome and ensemble, actually work out the person's ApoE status from the actual. This is partly why genomics data is sensitive. Each of us has a particular genome, and we have to control the data in a sensitive manner because it actually tells quite a lot about your physical health, your predisposition to particular diseases, which if you have life insurance, you would realize the life insurance companies would be very keen to get hold of this information on particular people because it gives them predictive power. Moving on to transcriptomics. So transcriptomics is all about measuring the messenger RNA levels. These messenger RNAs that are being expressed by cells. So it gives you information about how much particular genes are being expressed into messenger RNA that then produces protein. It's a very mature field now. We started to do gene expression analysis in the 1990s, and so it's 30 years old. We started to do it with these types of chips, spotted microarrays, and I'll talk about how these work because I think actually understanding these gives you a sense of how you can work with DNA. So these are spotted microarrays. There's very established databases, Gene Expression Omnibus from NCBI, Array Express from the EBI, and there's well-established formats in the field. MAGML is a microarray gene expression markup language format. Extensive open source software as well, like Bioconductor is the #1 research software used for genomic analysis and gene expression data analysis. That's the omics technology that we'll give much more detail about. It has been revolutionized over the last 10-15 years by next generation sequencing. essentially machines that can massively sequence in parallel, counting how many messenger RNA molecules there are in particular samples. One next generation sequencing machine is equivalent to 160 million Sanger machines. So this is what we used to use for sequencing only 20 years ago. And now we can replace 160 million of these machines with one of the next generation sequencing machines. So it shows the sort of power of genomics data analysis we have now. Hopefully David's described how Sanger sequencing works, and essentially the next generation sequencing machines, they essentially do Sanger sequencing but in a massively parallel format, so that they sequence millions of small DNA sequences all at the same time. So proteomics is the third type of omics, and this essentially is about measuring globally all of the concentrations of all the proteins in a cell or a sample. So this gives you an indication of the activity of different enzymes, for instance, in a metabolic pathway. You may say, well, doesn't the messenger RNA give you that? Because the messenger RNA is translated into proteins. So the messenger RNA does give you a good indication of what genes are active and what genes are not active. But this process is not simply linear. Some proteins are formed in inactive forms, and they actually have to have other processing to be made active. And we also want to know about what is the state of the protein. So it may have different levels of phosphorylation in the signaling pathway. So there may be active forms of the protein and inactive forms, and proteomics gives you this additional information. So the third type of omics technology is proteomics. This is where we measure the protein concentration under different conditions within samples. There are a number of experimental techniques like 2D page. So this is a 2D page of a sample, and what's happened here is the sample has been put onto the 2D page, and we have the molecular mass of the protein being separated out here, and we have something called the isoelectric point, the pH at which the protein is neutral, being separated out here. And you can see that it is separated out some of the proteins well. Each dark blob is a protein, and we need to work out from the position the particular type of protein, and then we measure the intensity under a circle to measure how much of that protein was in the sample. You can just see in here that there's the ApoE3 protein being measured here. So this is techniques not particularly good in terms of resolution. A much better technique is mass spectrometry, which breaks the proteins up into bits usually and measures the mass of those bits and how much of those bits to actually reconstitute how much of that protein was in the original sample. So these are all techniques, but they're not as advanced as genomics techniques. This is because all of the proteins have different properties. A bit of DNA, it has the same sort of physical chemical properties, and you just sequence it, whereas a protein, it could go from something which is very soluble to something which is a transmembrane protein and is insoluble, and it would be very difficult to work with. They all have different properties, which makes proteomics much harder than genomics. And the final omics technique that we'll look at is metabolomics. So this is about measuring the concentration of all of the metabolites or small molecules in a sample. So here's a metabolic pathway again. We have small molecules. It's the citric acid cycle or the Krebs cycle. We've got citrate, which has this molecular structure with carbons, hydrogens and oxygens, and then this particular enzyme converts this to isocitrate, and then it's converted to ketoglutarate, and all the way around in this cycle. And metabolomics would measure the level of citrate, isocitrate, and other metabolites. And proteomics would measure the level of all of the enzymes that are doing these conversions. So you can see how the different omics technologies interrelate. One of the key techniques for metabolomics is high resolution nucleic magnetic resonance, NMR. So this is a similar technique to sort of NMR body scanners that you see produce pictures of people's brains, but you're doing it on just a single sample, and you're doing it across lots of different frequencies. So you can actually identify different small molecules like lactate, creatine, alanine as one of the amino acids. And by measuring the area of the peaks, you can get an idea of how much of that metabolite was in the sample. So that's the main approach of metabolomics. So as we've been sort of indicating, that the idea of, for a while, has actually been to try and combine all of this information. So this was one of the first papers that in 2007, getting closer to the whole picture, where they took E. coli and they did transcriptomics, proteomics and metabolomics, and then they did lots of analysis and looked at the components and the interaction of the components, all of the different signaling pathways, the metabolic pathways, and they did a complete model integrating all of the experimental data and trying to predict what was going on in the cell in terms of metabolic fluxes of different metabolites going through the enzyme pathways. So this was ideally what you want to understand about a biological system. If you have this sort of understanding, then you know that if this enzyme gets a particular mutation which makes it inactive, you can actually work out how the system will respond to that, diagnose the problem, and potentially come up with some way of solving it. So it's a bit like having the blueprint for a car and trying to resolve some problem with the engine, rather than just currently at the moment in a lot of drug discovery, you just randomly try different targets and sort of hope that you hit something which explains a particular disease. So this is the more engineering-based approach to trying to understand systems. But unfortunately, we are probably not going to reach that level of understanding, that mechanistic understanding for human cells, eukaryotic cells, in the near future, because they're magnitudes more complex than bacterial cells. But in a way, we don't need to have that understanding to be able to apply machine learning in a useful manner, because machine learning can pick out patterns within the biological data. So we don't need to have a mechanistic understanding. I mean, it helps if we incorporate prior knowledge of what we do understand, but we don't need it to be a complete mechanistic understanding. I've used this paper, Khan et al. 2001, as an example of this. It's an early paper, but the earlier papers were simpler systems and they're easier to understand. The later papers are hugely complicated systems they're studying now. So what did this look at? Well, it looked at small round blue cell tumours. So this is a type of cancer, sarcoma, using gene expression data. And the whole purpose of the study was, given a particular cancer sample, can we determine what type of cancer it is? So small round blue cell tumours, these have all been different, they've been coloured differently, but they look very similar, and it's actually quite difficult to tell them apart from the histopathology slides. But there are different types: Ewing sarcoma, neuroblastoma, rhabdomyosarcoma, non-Hodgkin's lymphoma. And the idea of this paper was to identify them just from the gene expression data. So this was the procedure that they followed, and you'll recognize it as a sort of classic machine learning prediction approach. They only had 88 experiments, 88 patient samples. They used 63 for training, 25 as out of sample test samples, and they only had 6,567 genes. These are human samples, but this was 2001, before next generation sequencing, so they only were measuring 6,500 gene expression levels. So they filtered the genes initially. They filtered in terms of what genes actually do change. If the gene stays the same, it's not a very useful gene. If the gene has a very low expression, it may not be expressed, so it's not very useful. So they converted the 6,000 genes into 2,000 genes with 88 samples. They then used principal component analysis to do dimensionality reduction, and they actually only used 10 principal components. So this is one thing about gene expression data and other types of biological data, is that one of the aspects is that there's usually a lot of genes that are all going up and down at the same time. So Ewing sarcoma involves a chromosomal rearrangement which may cause a number of genes to all go down in expression or all go up in expression in that area of the chromosome. So we only get 10 principal components out of these 2,300 genes. They then did threefold cross-validation. They randomly partitioned the 63 training samples into 3 groups. They did 2/3 training, 1/3 validation, and they trained a neural network. So I don't think you even say that, is this a deep neural network? Probably not. This is actually an accurate schematic. It is a single a sort of 2-layer neural network, not even a hidden layer, an input layer and an output layer. The 10 gene expression, well, the 10 principal component values going in here, and 4 predictions of what type of sarcoma: Ewing sarcoma, rhabdomyosarcoma, neuroblastoma. And so they trained these models with threefold cross-validation. They did repartitioning to see how stable it was, and then they were ranking genes for sensitivity, minimizing the number of genes for the dimensionality reduction. So they did a number of steps partly because they were working with so few experimental data points, the 88 samples, 63 training samples. So they didn't really have the millions of data samples to train a deep neural network. But they actually got very good performance. So these were the 25 test samples, and these are the outputs of the neural network. So Ewing sarcoma, rhabdomyosarcoma, neuroblastoma. And you can see the first sample, 0.76 output for neuroblastoma. And then they used a method to judge, is this significantly bigger than the other outputs? And they determined, yes, it is. So they diagnosed it as neuroblastoma, and the histology actually told them it was neuroblastoma type C, so it was a positive match. Next one, Ewing sarcoma. Diagnosis Ewing sarcoma because it's very definite. It turns out to be Ewing sarcoma C. So the third one, you can see that it's rhabdomyosarcoma, but only just. It's only just narrowly better than this one, and only slightly better than this. So it wasn't significant enough. So they didn't diagnose it as anything, and they were correct not to diagnose it as one of these 4 types of sarcoma, because it turned out to be osteosarcoma, which is bone cancer. So they got very good results from this application of a very simple machine learning neural network to gene expression data. This is a sort of classic type of cancer subtype prediction using omics data. So they don't really know what's actually happening mechanistically, but actually this analysis allows you to dig into the patterns which might reveal what's happening. So Ewing sarcoma, if you looked at the principal components that are determining the sensitivity, the positive higher values of that output, what are the weights involved, then you'll see that this actually highlights the genes in the particular region of the chromosome which is responsible for the chromosome defect which causes Ewing sarcoma. So yeah, you can interrogate the machine learning techniques to get biological information out of them, but you don't actually have a full understanding of all of the mechanisms here. Okay, so I think this is probably maybe been a bit of a shock to the system for some of you coming from sort of machine learning, mathematics into this world of systems biology. But in summary, we've made an argument that most diseases often need a good understanding of complete biological systems, the components making up the systems, genes, proteins, metabolites, and also the interactions of these components in terms of how they work together in the area which is called systems biology. We've introduced a number of high throughput omics technologies, genomics, which gives us a parts list of the components of the biological system, transcriptomics, proteomics, metabolomics, which gives you a picture of the concentrations of the different components over different samples or over time that we saw in Streptococcus pneumonia. And we've also introduced a number of biological networks, signaling pathways, gene regulation networks, protein-protein interaction networks, metabolic networks, which really form the structure that allows you to give some idea of mechanistic interactions between the components. And that ideally would act as prior knowledge that you could put into the the omics data analysis. Okay, so that's the sort of biological aspects covered. In the next lecture, we're going to move on to transcriptomics and go in more detail into that particular omics technology, which is one of the most important.